Hello everyone, my name is Henry Chase Gaju from Rwanda. Uh, in this presentation titled Nanotechnology Rescues, I'll be explaining how cancer rises, how we can understand it in the context of today's diagnostic and therapeutic tools, and then how nanotechnology can supplement what we have today to improve the treatment and detection of cancer. Welcome. Yeah, the first part of this presentation is going to be understanding cancer. And throughout the presentation, I talk about de jure, which is Latin for by the law, breaching the rules, those which matter, cancer voyage, and then who takes the blame. Let's start. So understanding cancer requires to mention the notion of DNA and genes. So except a few cell types in the human body, all other cells have DNA, which is short for the deoxyribonucleic acid. And DNA is a structure which is double-stranded. This is the first strand, the second strand, and then it is joined by these red lines which represent the hydrogen bonds and to form what looks like a ladder. This is the structure of a ladder, and then coiled to form a double helix. So that's the structure of the DNA. And a gene is a portion along the DNA length which specifies a particular rule. So let's say from here to here is the first gene, from here maybe to here is another gene, and so on and so on. And the rules given by genes, either by making proteins or controlling other genes, determine a particular trait or body process, let's say skin color, body size, even cellular division. So when we say breaching the rules, those which matter, and understanding cancer in the context of cancer, it just means that cancer arises when particular rules are breached. In other words, when the rules governing cellular division or genes governing cellular division are destroyed. Then we introduce the concept of proto-oncogenes. Proto-oncogenes uh, literally means before becoming oncogenes, and then oncogenes is literally cancer genes. So the idea is that they can become cancer genes if they are changed, and those are the genes which code for growth factors, growth factor receptors, mitogenic xenotransducers, or any other thing related to cellular division. After cancer has been developing, there's a possibility that cancer can propagate to other parts of the body. There are two types of tumors, one which is the benign tumor. This one doesn't metastasize to other parts of the body, but the malignant tumors, they propagate to other body parts. How do we know they go? A primary tumor may form in lungs and metastasize to the brain. So if we analyze the brain tumor, we are going to find characteristic of lung cells and not of brain cells. So we know it came from the lungs. Metastasis is explained by the seven-step process called the invasion metastasis cascade, uh, which is starting by local invasion, intravasation or moving inside the vessels, circulation, arrest, extravasation, micrometastasis, and colonization. We explain this in detail. First step is that a tumor forms, you know. So a tumor forms, and the tumor cells plant themselves in the extracellular matrix and the stroma, and that is called local invasion. Second step is intravasation. So after these cells have been planted themselves in the extracellular matrix, then they find a way to enter the blood vessels or the lymphatic vessels, and they can do this by either stimulating angiogenesis, which is the formation of new blood vessels, or they can enter in the already existing vessels. The next step is then circulation. So they've entered now in the blood vessels and they are circulating. Some die, some do not die. And then on the fourth step, they are trapped in smaller capillaries because these cancer cells are way too big, having a diameter of around 20 micrometers, and they're not flexible. but the capillaries are having a diameter of 3 to 8 micrometers, so they're stuck. And it can be either in the brain, in the bones, in the lungs. So when they are stuck, it's called arrest. The fifth step is extravasation. So now after they are stuck, they found a way to move out from the blood vessels, the capillaries, into the tissues around that vessels. The way they do this is by obliterating the vessel wall. I mean, as they grow, they push away the vessel wall and they move out. Once in the tissue, they form the micrometastasis. That step is called micrometastasis formation, and those micrometastases are capable of growing into clinically detectable masses. The last step is colonization, which is forming new tumors, new masses in this new location. So that's how they move from one organ to another, metastasis. Let's now talk about who takes the blame. I mean, what causes cancer? We should mention that there are around a quadrillion. A quadrillion is like 10 million billions mitotic divisions occurring in a human lifetime. So, with such a great number comes high risks of getting cancer because each time there is cellular division, there must be DNA replication. And once DNA is being replicated, there's a chance that there can be change in those genes which deal with cellular division, and so cancer can arise. That's why there's a greater chance naturally. But then studies show that only 1 in 10 quadrillion mitotic division, which are a total of cellular divisions that can happen in lives of 10 individuals, only 1 in 10 quadrillion would directly or indirectly lead to clinically detectable cancer. So we would say actually that body uh, naturally beats cancer, right? However, if we consult another study of the global estimated number of deaths by cause from 1980 to 2020, we see that cancer, which is shown by this blue line on the graph, is killing many people than other fatal diseases, than HIV, AIDS, diabetes, and even hepatitis combined. It's mysterious. How is it that it's killing many people than other fatal diseases when actually the body can beat cancer naturally? So it just makes us think that there is something acting in opposition to the body's ability to resist cancer. And that same thing is carcinogens and mutagens. Carcinogens and mutagens are those chemical or physical species which come and change the chemical structure of the DNA, right? They can be ionizing radiations like gamma, X-rays. They can be chemical species like arsenic found in pesticides, or even chlorinated paraffins, which are used in plastics manufacture. They can be anything. So these kind of things, they come in the body and change the structure of the DNA, and by doing so change the structures of the proto-oncogenes, making them oncogenes, and then cancer start. And these things are usually integrated in our daily life, so we encounter them day to day. That's why cancer is killing many people today. So I hope that at this point it is understandable uh, how cancer comes, how uh, it voyages or moves to other parts of the body, and then what causes it in simpler terms. And that is the end of this part one, understanding cancer. Thanks for following. And the next video is going to be about cancer in the context of today's tools.